So welcome everybody. It's a great pleasure for me to introduce Professor Rick Relyea. He is working currently at Relyea Polytechnic Institute, and he has spent his career working to merge the fields of ecology, evolution, and ecotoxicology. So now he's focusing his work on the impact of pollutants on the interaction among micro or between micro and microorganisms. So Rick, I'll let you the opportunity to talk about that. Yeah.
Good morning, everyone. Good morning, everyone. Bonjour. I want to thank Stefan and Emily and the whole organizing committee for inviting me here. And thank all of you for getting up in the morning after a late night boat ride. I'm sure lots of you could have used a little more sleep. But I also want to sort of give you a warning. Why am I here? I'm not a microbiologist. And this is what happened a year ago. Stefan wrote to me and said, you know, we'd like you to be a keynote speaker. And I tried very diplomatically to say, I think you've got the wrong guy. I'm not a microbiologist. I'm an ecotoxicologist and an ecologist and limnologist, etc. But I really don't focus on the microbial level. We include it. We look at algae of all sorts, etc. But he said, my task here is to talk about what we do in our research group and hopefully maybe inspire you with the kind of things that we are able to do at the levels at which we work and maybe inspire you to think about these kinds of things in your own systems, because we do share a tremendous number of common interests. When we look at reviews of what people work on in microbial ecology and evolution, we really think about a lot of the same things. We think about competition, we think about predation, we think about pathogens, we think about mutualisms and cooperation. We think about all of the same things, but I think you'd agree we don't really talk to each other all that much, and we should. So what you'll see is I think about conceptually all the things that you think about, but with some different systems and some different benefits and I should say advantages and disadvantages in our system compared to the systems you work on. So let's talk about this. I'll focus on pesticides. We work with other contaminants, but we have a lot of work on pesticides that I want to tell you about. And if you know anything about historic pesticide use, here's what we used to do with DDT. We used to fog your backyard all over the world. So if you were, say, a migrant worker in the U.S. coming across Mexico, we'd fog your head to get rid of head lice. If you were on an international flight, we'd fog the cabin of the airplane. And we did that because we knew the benefits. And we knew that there were no costs. There were no downsides of DDT in the 1950s, right? It was the miracle insecticide. And we didn't know anything else that it did that was bad. And of course now we do, and we have a different perspective. We have to always think about why do we do these things, and we don't do them for no reason. We think about the benefits of pesticide use, whether it's increasing crop yields, saving children's lives. It's really important that we use this. But what we want to know is, what are the downsides? What are the unexpected, the unintended impacts of spraying pesticides or any other contaminant in this world? So we want to think about that. So I want to walk you through what we always think about is how do we traditionally do this and how we maybe should be doing this. So we traditionally do this looking at simple things like LC50 tests, where we're going to look at, on the x-axis, different concentrations of some contaminant and look at how well you survive or how well you perform in some way. And you almost always get a curve that looks something like this. And what we try to look for is the LC50, that concentration that kills half of you. So when there's none, nobody dies. When there's a lot, everybody dies. And somewhere in between is this magic number we look for, the LC50. And we know that we can compare different pesticides. We can compare sensitivity of different organisms. And that works really well. And the whole idea is that it's the dose that makes the poison. It's not the poison itself. It's how much of it you're exposed to. So in traditional toxicology, the way a lot of it's still done, is we think about these things in terms of protocols where we'll test some model organisms. So a lot of our tests have to use a few model organisms, things like a bird, a mammal, a fish, and an invertebrate like Daphnia, Zooplankton. We test them by themselves and we test them for a few days. Now some other tests will go longer than that, but the vast majority follow that protocol. We take a tiered approach where we might determine the LC50 in the lab. We might manipulate a few other things, pH, temperature, etc. We might even test a few concentrations in nature, but we typically do that. Species are alone and they don't interact with anybody. And what we want to know is what concentrations will kill you and what's the chance that you'll ever be exposed to that concentration. Because logically, you put those two things together, and if you're not exposed to a concentration that will kill you, the pesticide really doesn't pose a risk. Completely logical, and as you'll see, completely wrong. And it's wrong for some really good ecological reasons. And that's his other philosophy, what we would call community ecotoxicology. We have to think about the food web. You know, we have to think about these single species tests are a good starting place, but they're not sufficient because everybody lives in a community or a food web. So we have to think about that. If we go back to the original definition of ecotoxicology, people were talking about that. If we look at what Rachel Carson said, she was talking about that. We test individual species, but these things live in systems. And we have to think about what they do in the whole system because there's often unintended consequences, like this recent story. Just because you're out there to kill mosquitoes doesn't mean that's all you kill. We have to think about these unintended consequences. The same with pharmaceuticals and the unintended consequences that we call side effects. So you have to think about that. So here's the question I want to ask. If we bring in more ecology to ecotoxicology, how would our perspective change? And this is a question we've been working on for about 15 years and trying to put this ecology back into ecotoxicology to see how much our perspective does change. Now I want to assure you that your food webs are complex and so are ours. This is just a portion of a food web from a lake that we work in, more than 350 species so far that we monitor in that lake. It's complex. Now they're not all as tiny as yours, but some of them are really big and really hard to work on. But it is really complex. And what we try to do is break it down to understand who are the major players, who are the important players, and not take on the whole complexity here, but really think about functional groups, et cetera, in ways that we can kind of handle it and wrap our head around it a bit easier. We include all of the major players, and this is from decades and decades of people working on understanding who are the major players, the algae and the aquatic plants, a lot of the different consumers, both invertebrates and vertebrates, a lot of the major predators and omnivores, as well as some top predators. Depending on the experiment, we work on different players in the system. We do a lot of work with amphibians in particular because of a lot of indication that amphibians are declining around the world. In some places, it's correlated with the use of pesticides. So we think a lot about amphibians, specifically on tadpoles. We want to work on 4 different pesticides today. 3 of them are inhibitors of acetylcholinesterase. They typically slow down your nervous system. The other one is Roundup, and that, of course, is an herbicide that works very differently and is designed to kill plants, not kill animals. And everything that we collect, everything we work on, we collect from nature. We think that's really important. So all the organisms come from nature. These are some frog eggs that we pick up in the spring. And we know their history, and we know what they've been exposed to, at least in their lifetime. So we find that to be important. So now let's add some ecology. So here's the first one. Almost all the animals we work on live with predators, and probably most of the microbes you work with live with predators. And we want to ask, does that change the toxicology of pesticides? So when we think about this, we can raise these animals with different kinds of pesticides, different concentrations, and with and without the smell of predators in the water. So if you don't know, these kind of predators, this is a beetle predator eating a tadpole, of course, and all predators in the water give off a smell. And they give off a smell that's some combination of the smell of a predator and the smell of a dying prey, and it scares the heck out of everybody. If you're a relative, say, of this tadpole, if you're a closely related species. and it scares you and makes you do all kinds of things. It changes your behavior, it changes your morphology, it changes your life history, it changes stress hormones, it does all of these things. And we asked that a number of years ago, does it also affect how pesticides impact you? And no one had ever asked this question before because this is not a traditional toxicology question. We raise them one species at a time, and that's all we do. But we've been working a lot with predator cues and understanding how animals respond to predators, so we started asking, and this is just one of our experiments. These are bullfrog tadpoles, and first we'll show you how many animals die with either no pesticide on top or carbaryl, which we commercially sell as 7 in the US, and look at it over time. Right now there are no predator cues, and this is how many animals die without the pesticide and with the pesticide. This is, as far as everyone knew, a very safe amount of pesticides for tadpoles. We're going to add one thing. We're going to put a little predator in a cage. It's a salamander predator, and it can't touch you, but it can put off a smell that scares the heck out of you. And watch what happens. If we have just that smell of the predator versus we have it with the pesticide, it becomes about 50 times more deadly than we ever knew was possible when you combine the two together. We've done this with a number of different species. Everything you see, by the way, means it's standard errors. We've done this with a number of different species, and we find it's fairly common among insecticides, herbicides, across different species. And that tells us that we've really missed a lot of toxicity here, that the stress of predators makes pesticides way more deadly. They can make them way more deadly than we ever knew. What about competition? Your organisms and mine commonly experience competition. These are tadpoles in a drying up pond. They feel a lot of competition, really common interaction in nature. And we asked what would happen if animals exposed to competition and animals exposed to pesticides, whether that outcome would change. So let's take this case. These are bullfrog tadpoles again, how well you survive. And this is different concentrations of the herbicide Roundup. And we look at that, and this is how many survive when we have different amounts of Roundup. And you know, when you get up around 3 milligrams of active ingredient or acid equivalent of Roundup, a lot of animals die. But that's not the most interesting thing here. Is this is under low competition, medium competition, and high competition. And you see right in the middle what happens. You go from about 25% dying to about 75% dying. So 50% greater death simply because you have one more ecological interaction, competition. So we see that pesticides become more deadly when there's competition than we ever knew was possible. So reality number 3, we have to talk about indirect effects. All these organisms live in communities. It's not just pairwise interactions with predator and prey and one competitor and another, but they live in communities, and these communities have indirect effects. That is, just because you affect one, you may affect other members of the food web. And we've done a lot of work on this question, and the first one we'll talk about is Malathion. And Malathion, in the US, this is where we use it. Those are the crops we use it on. The favorite insecticide out there. Worldwide, probably number 2 to kill mosquitoes after DDT, to control malaria, sprayed directly over wetlands, as well as lots of areas that go into wetlands. What's nice about this is an old pesticide, but what's nice is it breaks down in a few days. It's also not toxic to most vertebrates, like tadpoles, at less than, say, 1,000 parts per billion. But it's really toxic to zooplankton because they're invertebrates, and most invertebrates are really sensitive to insecticides. That becomes important in a moment. But what would happen if you expose these vertebrates, these tadpoles, to way less than the LC50, like 1% of the LC50? And if you were traditionally thinking, what is the paradigm? 1% of the LC50, no animals will die. That is considered a really, really safe amount. So let's ask the question, is that true when we think about a whole food web? So let's do a thought experiment. We've got a simple food web. We have zooplankton eating phytoplankton, the floating algae. We have tadpoles that eat Periphyton, I should say biofilm in this group, but it's the same thing. It's the slimy algae that and fungi, et cetera, that's attached to rocks and logs, et cetera, and they compete for resources like light. So what happens? You have an airplane go over, it sprays the pond or the wetland, and what we know is that tadpoles won't die, but zooplankton are really sensitive. And if they're really sensitive, they go down, and the stuff that they eat is no longer eaten, and it should become abundant. And if it becomes abundant, the water should turn pea green, and it should block the light for the algae below it, the Periphyton. And if you block the light, it can't grow, and therefore the tadpoles shouldn't be able to grow very well, especially if the tadpoles experience competition. That's the thought experiment. Now we're going to ask, does it actually work this way? Here are the treatments. We have a control. We have a really tiny amount applied once a week, which is the frequency that may come into these wetlands. 5 times more, 25 times more with high or low competition of the tadpoles. And let's see what happens. This is a setup. These are big outdoor tanks. These are about 2 meters across, about 1,000 liters of water in each tank, and they're covered so you can control what goes in and what goes out. And we measure all sorts of things along the way. These experiments typically take 2 or 3 months. We look at how well the tadpoles survive and how big they are when they come out into a frog, and we slowly dry the tanks in August, which is when the ponds naturally dry where these animals have come from. So let's look at what happens to the zooplankton. We know they're really sensitive. No surprise, if you had any amount of insecticide, you knock out the vast majority of zooplankton. Whether it's high or low tadpole competition doesn't matter. It's toxic directly to zooplankton. But what else will happen? Here's what happens to the phytoplankton, the floating algae, and you'll see the more that you add, the more zooplankton you kill, the more the algae blooms. And here's just an image of that, and it just turns sort of pea green. And that happens with or without competition of tadpoles, doesn't matter, the water turns pea green. So here's the Periphyton, the algae below that the tadpoles do like. Small effects here under low competition, but under high competition, look what happens right here. A very tiny amount applied once a week knocks out an awful lot of the food that the tadpoles rely on. And the reason is it keeps getting shaded. Every time the zooplankton try to bounce back from the pesticide application, it gets applied again, and they keep getting knocked down. And it's in this disturbed state that you keep shading the food. And if you keep shading the food, what happens to these are leopard frogs. What happens is that their survival, well, there's not a lot of competition. They do okay, about 85 or 80% survive. But look what happens under high competition, where there's not enough food. These animals don't do well. About 43% of them die compared to the control, not because it's toxic to them. It's not. It's because you took their food away. And it's an indirect effect that shows us that we have to think about these things. We knock out the zooplankton, we favor the phytoplankton, that shades out the Periphyton, and the animals don't metamorphose. If you walked up to a pond like this that had been exposed, you'd see a drying pond with a lot of tadpoles swimming around. And ultimately, they will die from the drying pond, but they never would be there had Malathion not been in that system. And again, it breaks down in a few days. That Malathion is long gone. You wouldn't even find it there. But about half the animals would be dead because of that indirect effect. So that's what we see. About 43% of these animals die because of a chain of events that happens. And as importantly, it's that frequent tiny dose that did all the damage, not the dose that's 5 or 25 times bigger. So it wasn't the dose. It's the frequency of the dose that made all the difference to these animals. We've tried to reach generalities in all the work that we do to give some kind of rules of thumb, and we've done that in a number of follow-up studies to understand, for example, the interactions of the lethal predator effects and the non-lethal predator effects with Malathion. We've looked at whether we see similar things with other acetylcholinesterase inhibiting insecticides, and we do. The rules follow. The same exact thing happens. We look at different communities from the east coast of North America and the west coast of North America, and the same thing happens. So we have some great generality that when we add these insecticides, the same thing happens in different food webs with different insecticides, etc. So we have some nice generalities. Number 4, a lot of our animals live with plants, and none of these experiments I've talked about so far have anything to do with plants. So, one of my graduate students, Will Brogan, said, I wonder what plants would do, whether it would make a difference. We've got all these insecticides killing zooplankton. setting off this chain of events. But he came across some literature that said, you know, plants sometimes, in a very few studies, plants can take pesticides out of the water. They can absorb it onto their surfaces, and that's good. So we started looking at this with one plant, Elodea, and we asked what would happen to the zooplankton if we had different amounts of the plant. Very simple laboratory experiment, different concentrations of malathion, how well the Daphnia survive on the y-axis, and this is what happens if you don't have any plants. But if you add a few, or add a few more, you get really good survival. You go from none to about 80% of the animals surviving, simply because you added a plant. So he said, let's look at some other plants. This is what the experiment looks like. These are jars with soil and either no plants, or there's Elodea again. And he said, let's look at other kinds of plants. Can other plants do this too? And the answer is yeah. Every plant we look at can do the same exact thing. It doesn't matter what leaf shape you have. It doesn't matter whether you take more nutrients out of the water versus out of the soil. Every plant we look at can do this. And he said, well, just for fun, let's look at some things that kind of look like plants but aren't plants. So let's look at plastic plants, like an aquarium plant. Can't do it. So it's not surface area that matters. Let's look at some green rope. That can't do it. You've got to be a plant. And every plant that we looked at can mitigate the effect of the insecticide. And what we now know is with Malathion, it is not by plant absorption. And that confused us for quite a while. How is it? Because the only studies out there say it's due to plant absorption. What he discovered is that plants are altering the abiotic environment, and as many of you appreciate, plants in water, when they start photosynthesizing, they raise the pH. And they raise the pH, and at high pH, this pesticide breaks down orders of magnitude faster. So it's the photosynthesis of the plant, which is why every plant can do it. It raises the pH. And we know this because we can take the plant out and chemically raise the pH, and it acts exactly like the plant. So that's how we know it's pH and not absorption. Does this happen if you come out of the lab, away from just a jar, and go to a more natural setup? Here's what happens. He's got these outdoor mesocosms again. Again, these are 1,000-liter tanks. No plants, 10 plants, 50, or 100. And he said, what happens over long term if you have plants? Here's just an example over time. This is what happens if you have no plants and you add the insecticide. You get this classic algal bloom that we talked about before. The water turns pea green, the whole community is thrown off. But if you add even just 10 individuals, 10 shoots of Elodea, you come back a couple months later, and you know what you have? More plants. You have no algal bloom. So it takes very few plants to raise the pH and change the entire food web and really protect the entire food web from this insecticide, something that we didn't have any idea about. We now know from our most recent work that we actually have new rules of thumb, that we now know that some plants and some insecticides do work by absorption and others work by pH. And it turns out it's a simple rule of thumb. We have to know the KOW. We have to know the hydrophobicity of the insecticide. And things that are really hydrophobic absorb the plants, and things that are not are mitigated through raising pH. So we now have a nice rule of thumb to say all we need to know is the KOW value of the insecticide, and we will tell you whether it can be mitigated by plants and the mechanism by which it can be mitigated by plants, which we think is really helpful for people. So plants can mitigate insecticides. They can either take it out of the water or they can raise the pH to break it down. And it happens in the lab. It happens in these semi-natural experiments, and its outcomes are all predictable. So what does that tell us? We know the risk assessment can't be based on just the dose. It doesn't work. We have to think about things like stress from predation, stress from competition. We have to think about the presence of plants. We have to think about indirect effects. But it isn't that we have to throw our hands up. We are developing general rules of thumb to know how these food webs operate with these different insecticides and herbicides coming in. And that's always the goal, is to come up with generality. Now, I said we're going to put the ecology back into ecotoxicology. The last thing we want to do is think about putting the evolution into all of this. And I heard a lot of great talks on the evolution of microbes, and you know, that's to some degree a lot easier because of generation times. We want to talk about two organisms that have much longer generation times, maybe 10 days or 2 years. And can we get the evolution of pesticide tolerance is something that lives much, much longer than, say, microbes. Let's talk about zooplankton first. In the zooplankton case, one of the zooplankton that we work with is right here. And what we're looking for is natural patterns. So where do you see it? And particularly if you go close and far away from farms where pesticides are sprayed, do you see over the landscape any pattern that suggests they've evolved tolerance to the pesticides that are used? So we collect these animals from 10 different places. We raise them in 2 generations to get rid of what we call maternal effects. And then we test their tolerance to something like chlorpyrifos, really common insecticide. This is done in western Pennsylvania, which is up in the northeast corner of the U.S., and it's a really commonly used insecticide on all kinds of crops, including corn, etc. This is the expectation. As you increase the amount of agricultural land around the place where you collected them from, you should get an upsloping line that the ones living with more agriculture around them are more tolerant. And in fact, across 10 populations, it's exactly what we find. We can look at a 200-meter radius around that wetland. We can look at a 500-meter radius. It doesn't matter what we look at. They are more tolerant the closer they are to agriculture. We also see this in Daphnia, a really important player in aquatic communities among the zooplankton. So this is consistent with evolved tolerance to chlorpyrifos. But as I told you before, zooplankton play a really important role. They play such an important role in keeping the water clear, in preventing the phytoplankton blooming, and affecting everybody else, the periphyton, the amphibians, all the other grazers. So that's really important. So if you have a population that evolved tolerance, it should have a massive effect on the whole food web. And in fact, it does. As one of my students, RJ Bendis, we've got 200 mesocosms behind him where we look at the impact of evolved tolerance versus non-evolved tolerance. And what we see is, just as you would expect, when you have that one species that is typically really sensitive, if you evolve tolerance, that whole food web gets protected from that whole chain of events we've talked about. And if you don't, that whole food web is changed because of the pesticide. Not only because of that pesticide, but he also looked at cross-tolerance and other things that are working in the same way affecting acetylcholinesterase. You are protected against all of them. So that's a helpful piece of information. Amphibians, now we're talking about things that have a 2-year or more generation time. We think of that as much, much harder to ever evolve tolerance in the wild. So let's ask, what do we see? And the first work that we did a couple years ago, sure enough, found that the amphibians living closest to agriculture are more tolerant. So that was pretty interesting, but then it got more interesting. We see that they're cross-tolerant as well. But the most interesting thing that we looked at was whether this higher tolerance is evolving across generations, or can it be induced within a generation? There's only one example in the world we knew of in mosquitoes that says it can be induced. Like, we give you a little bit of pesticide and within hours or days, you're more tolerant to that pesticide. And we found that, but here's the key thing that we found. The induced populations were the ones that were really susceptible, far from agriculture. The constitutive tolerance was in these populations that were really tolerant, living near farms. And that made us wonder. If you looked at, these are amphibian eggs, and if you expose them to a little bit, we now raise them 15 populations, and we ask, what is the pattern that we see? Because if you've got inducible animals far from agriculture, constitutively high tolerant animals close to agriculture, that kind of sounds like the theory of genetic assimilation, which we'll mention here in a minute. Oops, I'm going backwards. There we go. So we're going to take tadpoles for a couple of days, give them a little bit of non-lethal insecticide, and then a couple of days later ask how well they do. How tolerant are they? This is what the theory tells us should happen. If you are far from agriculture in this case. We know that these animals are inducible. We think of them as the ancestral populations. They've been that way before farms were ever using pesticides. And over time, we think maybe this is what happens, that we get highly tolerant, non-inducible animals over generations and generations. And we ask, what does this really show us when we look at real data from inducible to constitutive? What do we see? And in fact, what we see is exactly that. These are the animals, these are the 5 populations farthest from agriculture, all showing inducible tolerance, less inducible tolerance, and on average constitutive tolerance. It seems to be that is the mechanism. But it really begs the question, how do you ever get inducible tolerance living far away from agriculture? How can you be inducible to something you've never seen, your population has never seen, presumably since before pesticides were invented? You're in a pristine forest far from agriculture. How is it that that happens? And this is how it happens. This paper is coming out in a few days. And it turns out that these animals, remember I told you they can respond to predators in all kinds of interesting ways. It turns out that your ability to respond to predator, which probably goes back millions of years, is being co-opted as also your ability to respond to pesticides. And we know that because if we scare the heck out of tadpoles for a few days, they become more tolerant to pesticides. So we know that it's a shared mechanism. And that's why these ancestral populations living far from agriculture have the ability to respond and become tolerant to pesticides because they're using the same mechanism. We don't know what that mechanism is, but they're using the same mechanism that they use for predators. So while it can and does evolve over generations, this tolerance can also be inducible in just a matter of hours or days. And if you have tolerance, you can change the entire food web in interesting ways. So the take-home message of all of this, what we've been trying to do is including ecology and evolution, as many of you do, to understand the impacts of contaminants in aquatic communities. The most effective way, and you've heard this message over and over, is to do this by looking at lots of different disciplines. We've done this in a number of different ways, everything from studying disease to phylogenetics to ecosystem ecology, etc. That's why we've been fortunate to discover so many different things. Also been fortunate to have a heck of a talented team over the last 15 years working on this, including a number of great undergraduate students who have worked with us. And of course, we're fortunate to have funding from the National Science Foundation, and that's our sole source. And I thank you so much for coming to the talk.